Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about the chapter 5 of your NCERT biology book, class 12th NCERT biology book, chapter 5, molecular basis of inheritance. We will discuss in details line by line. So stay tuned. At first, in this first paragraph, it is written that in the previous chapter you have learned the inheritance pattern and genetic basis of such patterns. At the time of Mendel, the nature of those factors which are actually gene regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of putative genetic material was investigated, culminating the realization that DNA, deoxyribonucleic acid, is the genetic material, at least for the majority of organisms. In class 11th, you have learned that nucleic acids are polymer of nucleotides. So, polymers are actually made up of many monomers. So, DNA or deoxyribonucleic acid is a polymer and the monomer of it are nucleotides. So, DNA or deoxyribonucleic acid and RNA or ribonucleic acid are two types of nucleic acids which are found in living systems. DNA acts as the genetic material in most of the organisms, and in some virus and DNA act as a genetic material in some virus, mostly function as messenger. RNA has additional role as well. It functions as adapter. In class 11, you have already learned about the structures of nucleotides and the way these monomer units are linked to form the nucleic acid and polymers. In this chapter, we are going to discuss the structure of DNA, its replication pattern, the process, then making the RNA from DNA in the process of transcription, the genetic code that determines the sequence of amino acids in protein, the process of protein synthesis in the process of translation, then the elementary basis of their regulation, the determination of complete nucleotide sequence of human genome during last decade has set a new era of genomics. In the last section, the essential of human genome sequencing and its consequences will be discussed. Let us begin our discussion by first understanding the basic structure of the most interesting molecule in the living system, that is DNA or deoxyribonucleic acid. In the subsequent sections, we will understand that why it is the most abundant genetic material and its relationship with RNA. So at first, the DNA. DNA is the long polymer of deoxyribonucleotides. Length usually defined as the number of nucleotides present in it. This is also the characteristic of an organism. For example, bacteriophage known as alpha into 174 has 5386 nucleotides, but the bacteriophage lambda virus has 48,502 base pairs. So it varies. The nucleotide sequence or the number of nucleotide sequence varies from organism to or species to species or from genus to genus. So what is the structure of polynucleotide chain? Let us recapitulate the chemical structure of polynucleotide chain. The monomer of this polynucleotide chain or nucleic acid is the nucleotide. It has three components: one nitrogenous base, a pentose sugar, and a phosphate group. There are two types of nitrogenous base. One is purine and second one is pyrimidine. Purine included adenine and guanine. Pyrimidine, cytosine, uracil and thymine. Uracil is present only in RNA. It is absent in DNA. But in case of RNA, instead of thymine, uracil is present. Cytosine is common in both DNA and RNA. Thymine is present in DNA Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to OH at 1 dash position of pentose sugar through N-glycosidic linkase to form a nucleoside such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or uridine. So when nitrogenous base only linked with the pentose sugar, it is known as a nucleoside. Only the pentose sugar and the nitrogenous base constitute nucleoside. But when the phosphate group is added to the pentose sugar and the nitrogenous base, it will be converted into a nucleotide. So only the pentose sugar plus nitrogenous base forms nucleoside. So in the place of that nitrogenous base, when adenine added to that place, it will be called as adenosine. When guanine, guanosine. When cytosine, cytidine. When uracil, uridine. When phosphate group is linked to the OH group of 5 dash carbon position of nucleoside through phosphoester linkase, a corresponding nucleotide or deoxynucleotide depending upon the sugar is present. When ribose sugar is present, it is known as ribose or nucleotide, or when the deoxyribose sugar is present, it is known as the deoxyribonucleic acid or nucleotide. Two nucleotides are linked through 3-5-phosphodiester linkase to form a dinucleotide. More nucleotides can be joined in such a manner to form polynucleotide chain. A polymer thus formed has one end free and a phosphate moiety at 5 dash end of the sugar, which is referred to as 5 dash end of polynucleotide chain. On the other hand, the polymer of sugar has a free hydroxyl group at 3 dash position, which is referred to as the 3 dash end of the polynucleotide chain. The backbone of polynucleotide chain is formed due to sugar and phosphate. The nitrogenous base is linked to sugar moiety projects from the backbone. So what is the basic structure and how it looks like? Suppose it is a pentose sugar. Pentose sugar, it have two OH group like this. It is a ribose sugar. Okay, it is a ribose sugar. So the phosphate group is added to this position, phosphate group, and nitrogenous base is added to this position. It is the case of ribose sugar. But when it is a deoxyribose sugar, which is present in your DNA, one O or one oxygen is less. Here at 2 dash position, this deoxyribose sugar lack one oxygen group from its 2 dash position. So it is the overall case of ribose and deoxyribose sugar. In RNA, nucleotide residue has additional OH group present at 2 dash position. Also in RNA, the uracil is found in the place of thymine. DNA has an acidic substance present in the nucleus, was first identified by Frederick Meissner. He named it as nuclein. However, due to technical limitations in isolation such as long polymer intact, the elucidation of the structure of DNA remained elusive for a very long period of time. It was only in 1953 James Watson and Francis Crick Based on the X-ray diffraction data produced by Wilkin and Franklin, proposed a very simple but famous double helix model for the structure of DNA. Then the Chargaff rule, according to which, and this Chargaff rule was proposed by Erwin Chargaff, and he told that there is a double bond or double hydrogen bond between adenine and thymine and 3 hydrogen bond between guanine and cytosine. The base pairing confers very unique property to the polynucleotide chain. They said to be the complementary to each other. Therefore, if one sequence of bases in one strand is known, then the sequence in the other strand can be predicted. Also, if each strand from a DNA, let us call as parental DNA, act as template for synthesis of a new strand. The two double strand DNA, let us call them as daughter DNA, thus produced would be identical to the parent DNA molecule. Because of this, the genetic implications of structure of DNA, the structure become very clear. The structure of DNA become very clear. So, if it is a DNA molecule, by using this strand as template, new strands can be formed. So, using this as parental DNA, new daughter strands will be formed. So, now the salient features of double helix structure of DNA are as follows. It is made up of two polynucleotide chain where the backbone is constituted by sugar and phosphate and the base or nitrogen bases projects inside. So these are ribose sugar, this is the phosphate which made the backbone of polynucleotide chain, and this nitrogenous bases projects inside. The two chains have anti-parallel polarity. It means one chain has 5' to 3' end, and one strand runs just opposite to it, that is 5' to 3'. The bases of two strands are paired through hydrogen bonds forming base pairs. Adenine form 2 hydrogen bond with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bond. As a result, guanine, always a purine, comes opposite to pyrimidine. This generates approximately uniform distance between two strands of helix. The two chains are coiled in right-handed fashion. The pitch of helix is 3.4 nanometer and there is roughly 10 base pairs in each turn. Consequently, the distance between a base pair is a helix approximately 0.34 nanometer. The plane of one base pair stack over the other in double helix. This in addition to hydrogen bond confers the stability of the helical structure. Then the proposition of double helix structure for DNA and its simplicity in explaining genetic implication become revolutionary. Very soon Francis Crick proposed central dogma in molecular biology, which states that genetic information flows from DNA to RNA, then RNA to protein. So it is the central dogma of life, which says that from DNA mRNA is formed by the process of transcription, and from mRNA protein is formed by the process of translation. DNA also formed itself, or from DNA another DNA strand can be formed or produced in the process of replication. This is the central dogma of life. Then in some virus, the flow of information is in reverse direction, that is RNA to DNA. So when it is RNA to DNA, it is reverse transcription. Then packing of DNA helix. DNA or the genetic material is so long, but our cell is too small, so it is packed inside the nucleus of the cell. Taken the distance between two consecutive base pairs, that is 0.34, because it is the DNA helix, and the overall length of it is 34 nanometer, and each strand have 10 base pairs, and between two base pairs the length or the distance between two base pairs is 0.34 nanometer. If the length of DNA double helix in a typical mammalian cell is calculated by multiplying total number of base pair with distance between two consecutive base pair, it is 6.6 into 10 to the power 9 base pair into 0.34, which is the distance, into 10 to the power minus 9 m per base pair. It comes out to be approximately 2.2 meters, a length that is far greater than the dimension of a typical nucleus. So the overall length is 2.2 meters, and it is very large than a cell or diameter of a cell itself. So it is packed inside a nucleus. In prokaryotes such as E. coli, though they do not have definite nucleus, DNA is not scattered throughout the cell. DNA being negatively charged, held with some protein in the region termed as nucleoid. The DNA in nucleoid is organized in large loop held by proteins. In eukaryote, the organization is much more complex. There is a set of positively charged basic proteins called histones. The protein acquire charge depending upon the abundance of amino acid residue with charged side chains. Histones are rich in basic amino acid residue lysine and arginine. Both amino acid residue carry positive charge in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pairs of DNA helix. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. Chromatins are thread-like stained, that means colored bodies seen inside the nucleus. Nucleosome in chromatin are seen as beads on the string. So beads on the string means this DNA molecule is surrounding the histone octamers. Then theoretically, how much such beads or nucleosome do you imagine are present in mammalian cell? The beads on string structure in chromatin is packed to form chromatin fiber that is further coiled and condenses at metaphase stage of cell division to form the chromosome. The packing of chromatin at higher level require additional set of protein collectively referred to as non-histone chromosomal protein. In a typical nucleus, some region of chromatin are loosely packed and stains light and referred to as euchromatin. The chromatin that is densely packed and stains dark are called heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive region. Then the search of genetic material. Even though discovery of nuclein by Meister and the proposition of principle of inheritance by Mendel were almost at the same time, but the DNA act as genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism of genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sudon, then Thomas Hunt Morgan and numerous other scientists had narrowed the search of chromosomes located in the nucleus of most cells, but the question that what molecule is actually the genetic material had not been answered. So at first, Frederick Griffith did the experiment with Streptococcus pneumoniae, the bacterium responsible for pneumonia, in 1928. He observed a miraculous transformation in bacteria. During the course of his experiment, a living organism, bacteria, had changed in physical form. So what he did? When Streptococcus pneumoniae bacteria are grown on a cultural plate, Some produce smooth, shiny colonies, while the other produce rough colonies. This is because S strain bacteria have a mucus or polysaccharide coat, while the R strain does not have it. Mice infected with S strain, that means virulent strain, or with shiny colony die from pneumonia infection, but the mice infected with R strain do not develop pneumonia. So when the mice is getting injected with the S strain, it will die. Then when it is injected with the R strain, it will not die. Griffith was able to kill the bacteria by heating them. He observed that heat killed S strain bacteria injected into mice did not kill them, but when he injected a mixture of heat killed S strain with R strain, the mice died. Moreover, he recovered living S bacteria from the dead mice. How it is possible? He concluded that the R strain bacteria had somehow been transformed by heat killed S strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat to become virulent. This must be due to transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiments. Then the biochemical characterization of transforming principle. Prior to the work of Oswald Avery, MacLeod and Maclean-McCarthy, the genetic material was thought to be protein. They worked to determine the biochemical nature of transforming principle. They purified from that transformed thing from the heat killed S cell to see which one could transform live R cell into S cell. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes and RNA digesting enzyme did not affect transformation. So the transforming substance was not a protein or DNA. Digestion with DNAs did inhibit the transformation. So he concluded that transformed thing is only DNA. Then the genetic material is DNA. The conformation experiment to prove the genetic material is DNA was carried out by Alfred Horsey and Martha Chase in 1952. So they work with virus that infect bacteria called bacteriophage. Bacteriophage attaches to bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as its own and subsequently manufacture more virus particles. Horsey and Chase worked to discover whether it was a protein or DNA, the virus that entered from the virus that entered the bacteria. They grow some virus in a medium that contained radioactive phosphorus and some on other medium that contained radioactive sulfur. Virus grown on presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains the phosphorus but protein does not. Similarly, virus grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Then radioactive phase were allowed to attach on E. coli bacteria. Then as infection proceeded, the bacteria coats were removed from the bacteria by the agitation them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with virus that had radioactive DNA were radioactive, indicating DNA was the genetic material that passed from the virus to the bacteria. Bacteria that were infected with virus that had radioactive protein were not radioactive. This indicates that protein did not enter the bacteria from the virus. DNA is therefore the genetic material that is passed from the virus to the bacteria. So it is their experiment where At first he radio labeled the phosphorus and the sulfur, then infection, blending, centrifugation. At the end of this experiment, they concluded that the DNA is the genetic material, and the DNA is the material which passes down from virus bacteriophage to the bacteria. Then the properties of genetic material DNA versus RNA. From the foregoing discussion, it is clear that debate between protein versus DNA as the genetic material was unequivocally resolved. From the Horsey's experiment, it became an established fact that DNA is the genetic material. However, it subsequently became clear that in some virus, RNA is the genetic material. For example, in tobacco mosaic virus, QB bacteriophage, answer to some questions such as why DNA is the predominant genetic material whereas RNA performs dynamic functions of messenger and adapter has to be found from differences between chemical structures of two nucleic acid molecules. Can you recall the two chemical difference between DNA and RNA? A molecule that can act as genetic material must fulfill the following criteria. One is it should be able to generate its replica. It should stable chemically and structurally. It should provide a scope for slow changes that are required for evolution. It should be able to express itself in the form of Mendelian character. If one examines each requirement one by one, because of the rule of base pairing and complementarity, both nucleic acid have the ability to direct their duplications. The other molecules in the living system such as proteins fail to fulfill the criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with changes in physiology of the organism. Stability as one of the properties of genetic material is very evident in Griffith transforming principle itself. That heat which killed the bacteria at least did not destroy some property of genetic material. This now can be easily explained in light of DNA that the two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, 2' group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. DNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among two nucleic acids, DNA is a better genetic material. In fact, the presence of thiamine at places of uracil also confers additional stability to the DNA. Both DNA and RNA are able to mutate. That means undergo mutation. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of protein. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. So, it is all about DNA and how DNA acts genetic material and how it was proved that the DNA is the genetic material. In my next class, I will discuss about the RNA world. Then how DNA replication occur, then how transcription and translation occur. So stay tuned and enjoy my videos like this. and please like the videos, share with others and subscribe to my channel Sunanda's Tutorial. Thank you.